Complement and its action. A very good day to all the listeners and viewers. The topic of discussion here is complement and its action. To learn about complement system and its role in immunology, it is very much necessary to get accustomed with the term complement. So, let us look upon first to concept of complement. Complement, a group of serum proteins which can be activated by antigen-antibody complexes or other substances which may result in lysis of a microbial target or a variety of other biological effects important in both innate and adaptive immunity. The majority of these proteins are produced by liver. Let us start with the history. In the late 19th century, a researcher named Jules Bordet, following the earlier research of Richard Peffer, was investigating the lysis of bacterium cholera vibrio, that is the agent which causes cholera, by immune sera, and found that the ability of an immune serum to lyse its target was lost upon heating it at 56 degrees Celsius for 30 minutes. This ability to cause lysis was also lost after simple storage of the serum for a few days at room temperature. Bordet showed that such heating did not destroy the antibodies. However, since the addition of a small amount of normal non-immune serum to the heat inactivated antiserum fully restored its capacity to specifically lyse cholera targets. Thus, the ability of immune serum to lyse bacteria depends not only on antibodies specific to C. vibrio, but also on a non-specific heat labile substance found in normal serum. These substances became known as complement since it complements the activity of the antibodies which are still present in heat inactivated antisera. Other scientist Paul Ulrich showed two components of the antiserum: heat stable, that is responsible for specific antibody, and heat sensitive, that is responsible for the lytic activity known as complement. Let us now look to the importance of complement system. Importance of complement can be highlighted as major effector system of the humoral IR, component of the innate, that is non-specific immune IR, results immediate response, and connection to the specific IR, where IR stand for immune response. Components of complement system involves inactive factors, cell surface receptors, and regulatory proteins. Talking about each one, first, that is inactive factors are present in the serum and body fluids which can activate each other in an enzyme cascade. Cell surface receptors, denoted as CR, are responsible for binding the activated complement components, whereas regulatory proteins are both soluble and cell surface bound. They are necessary to prevent uncontrolled complement activation. Let us now preview the various biological effects which can be attributed to the action of complement and identify those complement components or complexes which are responsible for these effects. Biological effects of complement. Biological effects of complement can be discussed under headings such as cytolysis, anaphylotoxin activity, chemotaxis, opsonization, and tissue damage. Let us see cytolysis first. It can be defined as destruction of target cells by lysis of the cell membrane. This is termed cytotoxicity in the case of nucleated cells, hemolysis for red blood cells, or bacteriolysis in the case of bacteria. Let us see anaphylatoxin activity. Stimulation of mast cells to release histamine and other substances, resulting in increased capillary permeability and local accumulation of fluid in the tissue. Now, let us see chemotaxis. It is attraction of polymorphonuclear neutrophils denoted as PMNs to a local site of inflammation. Whereas, let us see opsonization, facilitation to phagocytosis by macrophages or PMNs via cell surface receptors specific for complement components. Complement component receptors or CRs is called opsonization. Let us now have a look at tissue damage. Both the lytic complex and the inflammatory PMNs can cause considerable damage to normal tissues, for instance in an arthrous reaction or in immune complex disease. Moving ahead, let us now discuss the major functions of complement system. After initial activation, the various complement components interact in a highly regulated cascade to carry out a number of basic functions, including lysis of cells, bacteria and viruses, opsonization which promotes phagocytosis of particulate antigens, binding to specific complement receptors on cells of the immune system, triggering specific cell functions, inflammation and secretion of immunoregulatory molecules, immune clearance which removes immune complexes from the circulation and deposits them in the spleen and liver. Now, let us have a look at pathways of complement activation. Pathways of complement activation. The complement activation can be divided into three pathways: classical, lactine, that is mannose binding protein, and alternative, all of which result in the activation of C5 and led the formation of the membrane attack complexes, that is MAC. The three pathways differ in the way C5 is broken down, but after that the formation of MAC is essentially the same. Classical pathway. The classical pathway is triggered primarily by immune complexes containing antigen and immunoglobulin G and immunoglobulin M in the presence of complement components 1, 4, 2, 3, calcium and magnesium cations. They react in the order C1q, C1r, C1s, C4, C2, C3, C5, C6, C7, C8 and C9, while IgG1, IgG2 and IgG3, that is the most effective, can activate complement. IgG4 is not able to activate at all. C1 is the first complement component to participate in classical pathway. It is composed of C1q, C1r and C1s. Binding of C1q to antigen antibody complexes results in autocatalysis of C1r. The altered C1r cleaves C1s, and this cleaved C1s is capable of cleaving both C4 and C2. Activated C1s enzymatically cleaves C4 into C4a and C4b. C4b binds to the antigen bearing particle or cell membrane, while C4a remains in a biologically active peptide at the reaction site. C4b binds C2, which becomes susceptible to C1s and is cleaved into C2a and C2b. C2a remains complexed with C4b, whereas C2b is released. C4b2a complex is known as C3 convertase. C3 convertase in the presence of magnesium cleaves C3 into C3A and C3B. C3B binds to the membrane to form C4B 2A3B complex, whereas C3A remains in the microenvironment. C4B 2A3B complex functions as C5 convertase, which cleaves C5 into C5A and C5B. Generation of C5 convertase marks the end of the classical pathway. C5B initiates the formation of membrane attack complex. C1QRS can also bind to a number of agents including some retroviruses, mycoplasma, polyinosinic acid and aggregated IgG and initiate the classical pathway. Another pathway is alternative pathway. Alternative pathway is so called because it bypasses the requirement of antigen antibody complex C1, C2 and C4 components. It begins with the spontaneous activation of C3 in serum and requires factors B and D and magnesium all present in normal serum. A C3b like molecule, that is C3I, is generated by slow hydrolysis of native C3. C3I binds factor B, which is cleaved by factor D to produce C3IBB. C3IBB cleaves native C3 into C3A and C3B. C3b binds factor B, which is again cleaved by factor D to produce C3bBB. C3b has very short life and unless stabilizes by membrane or molecule present on many pathogens, it is quickly inactivated. In the absence of such a molecule, it binds quickly to RBCs via the C3b receptor denoted as CR1 and decay accelerating factor denoted by DAF prevents the binding of factor B. Another serum protein, factor H, can displace factor B and bind to C3b. Binding of factor H makes C3b more susceptible to factor 1, which then cleaves it into many fragments, that is IC3b, C3d, C3e. C3 convertase generated in the classical pathway is also regulated by DAF, CR1 and factor 1. Certain bacteria of their products such as peptidoglycan and polysaccharides can stabilize C3b. Thus, C3b bound to such a surface becomes relatively resistant to the action of factor 1. Binding of another protein, properdine, further stabilizes this complex. Hence, the alternative pathway is also known as the properdine pathway. Stabilized C3 convertase cleaves more C3 and produces C3b, BB, 3b complexes known as C5 convertase, which cleaves C5 into C5A and C5B. C5b initiates the formation of membrane attack complex. The alternative pathway provides a means of non-specific resistance against infection without the participation of antibodies and hence provides a first line of defense against a number of infectious agents. Some of the microbial components which can activate the alternative complement cascade include lipopolysaccharide denoted as LPS from gram-negative outer membranes, teichoic acid from gram-positive cell walls, certain viruses, parasites, heterologous red cells, zymosin from fungal and yeast cell walls and some parasite surface molecules. Aggregated immunoglobulins, particularly IgA, cobra venom factor, that is CVF, and other proteins for example proteases, clotting pathway products, also can activate the alternative pathway. After discussing about classical pathway and alternative pathway, the other one is lectin pathway. C4 activation can be achieved without antibody and C1 participation via the lectin pathway. 3 proteins initiate this pathway, namely a mannan binding lectin protein, that is MBL, and 2 mannan binding lectin associated serine proteases denoted as MASP and MASP2, all present in normal serum. MBL binds to certain mannose residues on many bacteria and subsequently interacts with MASP and MASP2. The MBL-MASP-MASP2 complex is similar to antibody C1QRS complex of classical pathway and leads to activation of C4, C2 and C3. The rest follows as in the classical pathway. Formation of MAC. C5 convertase generated by any of the pathways described earlier cleaves C5 into C5a and C5b. C5b instantaneously binds C6 and subsequently C7 to yield a hydrophobic C5b6-7 complex which attaches quickly to the plasma membrane. Subsequently, C8 binds to this complex and causes the insertion of several C9 molecules. The insertion of membrane attack complex causes formation of hole, that is 100 angstrom in the membrane, thus lysing the cell. If complement is activated on an antigen without a lipid membrane to which the C567 can attach, the C567 complex may bind to the nearby cells and initiate bystander lysis. Significance of complement activation. First, that is lysis of cells. The MAC that inserts itself into the membrane causes the cells to lyse. It may also cause the bystanding cells to lyse. Gram-positive bacteria, which are protected by their thick peptidoglycan layer, bacteria with a capsule or slime layer around their cell wall, or non-enveloped viruses are less susceptible to lysis. Second, that is inflammation and phagocytosis. C2b generated during the classical pathway of C activation causes vascular permeability and edema. C4a, C3a and C5a are all anaphylotoxins which cause basophil or mast cell degranulation and smooth muscle contraction. C5a and MAC are both chemotactic. C5a is also a potent activator of neutrophils, basophils and macrophages and thus amplifies the inflammation. It also causes induction of adhesion molecules on vascular endothelial cells and hence promotes diaphysis. C3b and C4b acts as opsonins on the surface of microorganisms and promote phagocytosis. Complement also helps to rid the body of antigen-antibody complexes. C3b receptors are present on RBCs which pick up complement coated immune complexes and deliver them to the Kupffer cells in the liver. This is called immune adherence. The classic component pathway appears to be important in the dissolution of immune complexes. Certain viruses are neutralized after interaction with antibody and complement components. C2a can be converted to C2 kinin which regulates blood pressure by causing blood vessels to dilate. They contribute to type 2 hypersensitivity, that is incompatible blood transfusion, hemolytic disease of the newborn, autoimmune hemolytic anemia and hyperacute graft rejection, and type 3 hypersensitivity, which includes Arthur's reaction, serum sickness, post-streptococcal glomerulonephritis, autoimmune disease and rheumatoid arthritis.
Summary of the complement system. The complement system comprises a group of serum proteins, many of which exist in inactive forms. Complement activation occurs by the classical, alternative or lectin pathways, each of which is initiated differently. The three pathways converge in a common sequence of events that leads to generation of a molecular complex that causes cell lysis. The classical pathway is initiated by antibody binding to a cell target. Reactions of immunoglobulin M and certain immunoglobulin G subclasses activate this pathway. Activation of the alternative and lectin pathways is antibody independent. These pathways are initiated by reaction of complement proteins with surface molecules of microorganisms. In addition to its key role in cell lysis, the complement system mediates opsonization of bacteria, activation of inflammation and clearance of immune complexes.